This is Making a Scientist, the podcast by young scientists for young scientists, featuring cutting-edge science and all of the life and work advice that you'll ever need to succeed. It's brought to life by brilliant scientists, and it's hosted by me, Alex Ainscough. We made it all the way to the series finale, and what an episode this is! This week, my guest is Dr. Lucie Low, who is a scientific program manager of the Tissue Chips for Drug Screening program at the National Institutes of Health, which encompasses programs of work such as the Tissue Chips for Disease Modelling and Tissue Chips in Space. Organs on chips, or tissue chips, are some of the hottest tools in the biosciences right now. In this episode, find out what they are, how they have immense potential to deliver huge benefits for patients, and to alleviate some of the ethical concerns of animal research by offering an alternative and perhaps a better way of testing new drugs, cosmetics, or even running clinical trials without causing any harm to animals or humans. Lucie and I also discuss how organs on chips are being used in experiments carried out aboard the International Space Station and some of the challenges associated with carrying out biological research in space. We'll also hear all about Lucie's career path, how she made the decisions that she did, and hear her incredible advice for early career researchers. I know I say this every time, but this really is a truly brilliant episode. Let's begin! Dr. Lucie Low, good morning. Thank you very much for joining me this morning. It's a pleasure to have you on the podcast.
Good morning, Alex. Thank you so much for inviting me to be here. It's lovely to meet you and looking forward to chatting.
Yeah, it's going to be amazing. So I'm so enthusiastic about this topic. Organ on a chip has been my life for the last 5 or 6 years, and I've just been so all consumed by it. So to talk with you is honestly is a pleasure. So I'd like to dive right in with organs on chips.
Yeah, let's do it.
So very first question then, what are organs on chips, and where did the concept come from?
So if you've been working on this, then you undoubtedly know yourself, but I'm happy to try and explain
please.
from my end as well. So organs on chips are little bioengineered devices that contain human cells, or in our case, we choose to work mainly on human cells, in little bioreactors fed by microfluidic channels, and they recreate the structure and function of human organs and tissues. So basically, they're these little devices that you can put cells in a certain kind of architecture with the right kind of support materials. You can feed them with media that contains all the kinds of growth factors and things that they need, and you can just basically sort of let them do their thing. It's like a little home away from home, and they replicate function of your organs and tissues in ways that it's just really difficult to recreate in other kinds of models right now. So there's all different kinds of ways of modelling human organs and tissues, but these microphysiological systems, or organs on chips, are these 3D style systems where they essentially create tissues that are a bit more true to life, to the ones actually inside your body, than say if you were to put them in a petri dish or if you were looking at animal tissues. So we like to think of them as slightly more realistic models of human tissue. In these little bioengineered devices, that means that we can look at different drugs and diseases and really sort of try and get some better models, if you like, of human tissues and human diseases.
So it's such a fascinating concept. Where's it come from? How did organ on a chip come about?
Well, I feel like it was one of those ideas that was such a great idea that someone would have come up with it at some point anyway. But certainly I work at the National Institutes of Health, and we like to think that we had a bit of a hand in helping spur the development of them.
Of course.
So, so essentially though, the first sort of the archetypal, if you like, organ on a chip came out from an idea from Don Ingber's lab, who was at Harvard and is at the Wyss Institute, and one of his postdocs, Dan Herr, and he created a chip that essentially recreated lung function, and they did this by putting the sort of the airway cells in this little device, and then they added biomechanical forces by stretching the membrane that the cells were sitting on by applying a vacuum that ran in a channel down the side. And so it was such an elegant idea, this kind of lung on a chip model. And it was one of the very first organ on chip models that came out. But at the same time, lots of other people were starting to think about how can we recreate sort of organs and tissues in a 3-dimensional organization, and how can we do that? So with advances that came in microfluidics and with 3D printing and microfabrication and bioprinting. Over the last 10 years or so, this field has really kind of grown. So the kind of that first chip was published in the literature. It came out in Science in 2010. But then the FDA and the NIH were like, oh, we obviously knew what kind of research was going on, but we're like, this is a really great way to look at how we can use these kinds of models to see potentially in the drug development process and see how we can advance regulatory science, basically. So the NIH and the FDA got together and funded some programs, and then that's basically kind of how it all kicked off. And then the NIH is still funding programs, 10, 12 years later. FDA is doing a lot of work. DARPA as well, the Defense Advanced Research Projects Agency here in the US, has done lots of research funding for organs on chips as well. And so the field's just really exploded over the last 10 years.
It really has. So something that I always find in the literature is that there's often a wording issue potentially, and a lot of people say there are things like organs on chips or tissue chips. So would you mind just explaining what the difference between a tissue and an organ chip is?
So honestly, there isn't really any. It's definitely just they're all kind of synonymous. So there's one of the things that we do talk about in the field is what should we call these things, and pretty much everyone is like, well, I'm going to call it what I want to call it, and then we'll just have to sort of work out how to get along. So the official term that NIH uses is microphysiological systems, which is a very broad term. We also talk about organs on chips. We talk about tissue chips. They're all pretty much the same thing. Within that broad name, though, there's lots of kind of different levels, if you like, of complexity. So some chips can be quite simplistic. They might just contain balls of cells or organoids in microfluidic platforms. So these cells in a ball are still feeling sort of some of the stretch or some of the shear forces, the biomechanical forces as fluid flows past them. But then you might get much more complicated chips where specific cell types are actually placed in very particular areas that are designed very much to recreate the architecture of your organ. So, you might have cells in a liver chip that are subject to different amounts of oxygenation as it comes through the media. So you're essentially creating an oxygen gradient within a liver chip. So there's lots of different ways that people can create and replicate the different kinds of internal milieu of your body. But the names are pretty much all synonymous, and they're all kind of meaning the same thing.
Yeah, sure. It's interesting because, like you say, I think everybody's just like, it's a bit of a free-for-all, right? Like everyone will just do what they do. Yeah. And there doesn't seem to be so much direction. But I think microphysiological systems, personally, I think that is a better all-encompassing term. And it also sounds pretty cool as well.
We say microphysiological systems or organs on chips, and people are like, oh, organs on chips, yeah.
Yeah, I suppose it's become such a big buzzword. And I mean, we can talk all about future in a bit, but I think that the future of organs on chips is truly fascinating. But what I want to focus on just right now is what are the biggest challenges or the major limitations that are associated with organ on a chip technology right now, as you see it? Yeah.
So it's new technology, right? So with any new technology, a decade sounds like a long time, but in biological, bioengineering terms, it's still very young, essentially. There's a lot of challenges that people still need to work out. So one of the big ones is what cells do you put in there? So with new technologies like stem cells, induced pluripotent stem cells, they've been around for about a decade or so. Well, Yamanaka, who helped create the first protocols, he got the Nobel Prize for it in 2012. So it's less than 10 years since that kind of technology has been around. So, we very much encourage our researchers to use stem cell technologies to put on their chips, because then they're using essentially renewable sources of cells that can be taken from a patient or a donor and then created on a chip to make like a U on a chip. Talk about precision medicine type efforts. But stem cell technology is still young. It can be really difficult to get the kinds of cells that you want. The differentiation protocols might not be very good. It might create tissues that are functionally very immature. So if you're creating, say, neural cells and you're trying to look at neurodegenerative diseases, you might get neurons that are actually very fetal in nature. They're very young. Or they don't last very long on tissue chips. So they might only last a few days and then they might start doing all kinds of crazy things. And so it can be very difficult to look at long-term disease pathology or repeated exposures of different kinds of drugs and things like that. So stem cells have this phenomenal promise, but it's, it depends on the tissue you're using. It can be really tricky. So, there's all kinds of pros and cons with all the different cell sourcing things.
So whilst all of these stem cell protocols are being refined and developed, what should organ-on-a-chip researchers be striving to use in these chips? Should they be looking more to use primary type cells because that's what we would get from somebody, or would you look more to use potentially cells that you would like a cell line more, something that's more robust? What's your feeling on it?
So it's not a very helpful answer because it really depends on the question you're asking.
Yeah.
So if you're looking at cancer on a chip and you can recreate what you need to show with a cell line that you can buy from, Lonza or wherever, and you can ask the question that you need to do and ask it in a reproducible and reliable way, then use the cell line that works. Okay. If you're looking at something else that requires a certain disease phenotype, then you might be having to look at primary cells or from donors or something like that. And then if you can make your model with those cells and get reliable results that replicate what they're supposed to replicate, it does what it says on the tin, if anyone remembers that advert, then, that works too. So like I said, we very much encourage our researchers to push towards the use of IPS because we want to help that technology mature as well. But it really depends on the question you're asking. And this is going to we come back to this over and over again in the tissue chip field is, what's the context of use of your platform? So, if it's all very well to create this lovely little model of something, but if it doesn't do what it's supposed to do in a reliable way and answer the questions that you need to ask, then it's not a very helpful model. So it's what people really need to do is think about the context of use of their model and then work out what's the best way to do that scientifically at this point. So that's totally kind of how we focus on it. We say, do what the science needs you to do. If you need to use a mixture of cell lines and primary lines and, IPS cells, then do it if you get a good model out of it.
No, definitely. And I think as well, it might be worth mentioning about how organ-on-a-chip is kind of like a tool, this technology. So, if you don't necessarily need to go through all of the hassle of, making and setting up your chips, because a lot of people fabricate their chips in-house, and you could just do this in a little dish if you're looking at, expression of something, then you don't need to go through it. So I suppose, that for me as well, because that's the big bottleneck is the fabrication of chips.
Yeah,
so
absolutely.
That's a big thing.
Absolutely.
And everybody makes it differently, which I find is also a little bit there's no sort of like unified way to do it, but it's one of those that if there was a unified way to do it, I'm sure there's somebody who's going to be turning a profit of it, and then it becomes who's going to be that person. And this is why everyone's got their own different protocols, and it's really like it makes it quite difficult to find out which one to use.
It does, and that actually is the exact, that you just hit on another challenge, which is this, there's so much difference between how people make them, which cells they use, all of that kind of stuff. It can be very difficult for standardization. It can be very difficult for commercialization. And so that's definitely something that everyone in the field is working for, so that, we can get to a stage where we've got all these different kinds of chips that we can send to the FDA and say, hey, here's the data from this chip. And they'll be like, oh, yes, we know this one. So. Yeah,
for sure. So, winding it back to something a little bit more simplistic, what's your favourite type of organ-on-a-chip device?
So I'm not going to pick favourites.
Okay, fair.
I love all our chips equally. So we have about 50. So I manage a program where we have about 50 or 60 different projects, and every single platform is slightly different. And special in its own way. So no, I actually really enjoy working with all of the different kinds of chips, although I am a neuroscientist by training. So I will just say that anything that's got neurons on, I'll always listen in a little bit more I see.
Fair fair enough. So you've already touched on iPSCs. But I wanted to maybe ask this question in a slightly different way. So I want to really understand how you think we could realize the blend of personalized medicine organs on a chip, because I've heard from a lot of people before how the revolution in genomics is going to be the way forward, and we're already seeing it with COVID, and how this next generation sequencing is also like something that's done by default with PCRs. So in addition to iPSCs on a chip, do you think how can we translate it from something that's done in a lab environment where you've got, you know, a green fingered lab assistant who's particularly able to get this organ on a chip running, to something that could be routinely used in hospitals? Because, you know, you want to have this like selection of the best drug for the best person, right? Like, you know, you want to have the selectivity. How do you think we can realize this? I know it's a pretty difficult question.
Yeah, it is a really great question, because it is a really difficult question. So I think in terms of when you're thinking about personalized medicine and precision medicine, and you said like in a hospital context, and you start thinking about clinical trials, then you have to be focusing on certainly iPS cells to populate at least certain relevant portions of, you know, your chip, your cells in your chip, because that's how you get patient-specific responses. Yeah. But then you also need to be working on creating a platform that technologically is very reliable and very robust, and doesn't necessarily need a green-fingered lab tech, like you said, or, you know, we like to joke and call them chip whisperers. You know, you need a technology that doesn't need a chip whisperer just to make it work. So
I have to steal that one in future presentations.
Oh yeah, please do. But this is one of the challenges, is that at the moment the tech can be very involved, and it can require quite great in-depth expertise to be able to make them work reliably. And so, you know, there really is going to be a push in the field towards creation of systems that are more automated. They might be therefore a bit more simplistic, so that they don't require, you know, 8 years of lab experience to be able to get something up and running. But you know, we really do see that there's going to be this need for creations of systems that are just complex enough, that can be automated to a certain extent, so that they can be translatable in any situation, and not just in a hospital, you know, lab, but also maybe on a battlefield, or you know, maybe out in remote areas of Africa, or, you know, even in space, you know, for astronauts in future. So, you know, the kind of the push towards creation of automated systems that are a little bit, or are fit for purpose, but just as complex as they need to be, I think is going to be a huge deal. But actually, one of the things the NIH is doing is actually funding a program right now called Clinical Trials on a Chip. And this is very much addressing the question you just asked. And then it's not that we're suddenly going to be running clinical trials on chips, but it's that, as you said before, chips are tools. They're tools in the process of trying to understand disease, trying to develop drugs. And so we want to see how they can be used in the context of decision making during a clinical trial. So, for example, if they can be used to help stratify patient subgroups. So if you're looking at a new chemotherapy or combinations of chemotherapies, if you can test on chips and work out what kinds of patients are going to respond better to which kinds of combinations, then you already have your patient stratification set before you even start a clinical trial. So there's all these opportunities to do kind of almost first in human style experiments on chips before they go into humans, and I think that's also going to really help push that personalized medicine aspect.
Yeah, that's absolutely fascinating, and I suppose you'd even use the same people, right? You'd for the clinical trials, it's the it's your response beforehand. So that's absolutely.
And it's another way to validate. It's another way to validate the chips as well, because if you're getting clinical trial data and then you can do the same experiment or test the same drugs on the tissue chip, you're seeing very clearly how well the tissue chip actually replicates and recreates that individual person's response, which is going to feed in towards the validation efforts we were talking about.
Yeah, for sure. So this is another like really fascinating part of the field, which I think is, it's just starting to take off. There've been a few publications on it, but it's the idea of the body on a chip, or the whole body on a chip, and this is where you would link lots of different organ on chips together, and you would usually do so via a common medium that sort of mimics blood, if you have connections via vascular type channels. But if you wanted to run a clinical trial on a chip, would it be just looking at somebody's liver rather than the whole body on a chip? because it's going to be, again, quite difficult to, get everything all primed at the same time and ready to run the body on a chip type experiment. So how would the clinical trial on a chip work?
So I'm going to go back to that point of it depends on the question you're asking.
Okay.
Again. So if you're looking at bone cancer, but what you're really interested in is how toxic this combination chemotherapeutics is for, liver, then you might want to link systems, but they might be quite simplistic systems, or, you might even not necessarily need to physically link them, but you could just take the media from one and then put it in a liver chip and see how it affects it. So there's lots of different ways you can address that question. But I would also just point out that I mentioned DARPA had been involved in funding over the years, and they actually funded the development of a 10-organ body on a chip system. They gave large grants to two what they call performers in the DARPA realm, and those labs actually created 10-organ linked systems. Don Ingber's group at the Wies was one of them, and he created the system that they call the Interrogator, which is terribly exciting.
It's a fierce
name. It is very fierce, yeah. And Linda Griffith at MIT created their own 10-organ system. But the stuff that's coming out from them now, and there's sort of this move towards commercialization of those systems as well, suggests to me that there is going to be commercially available linked multi-organ systems over the coming years that will be very, very informative for some of these questions that you were talking about, like when you're trying to set up a clinical trial. Like, you need to know what effect does the drug have on the actual disease it's supposed to be working on, but does it create toxic metabolites as it goes through liver metabolism? Does it create something that's going to damage the kidney and cause kidney failure? Does it create something that crosses the blood-brain barrier and then, kills all the neurons? So these are really important questions that you can ask on a chip, and that's why animal studies can be so helpful, is because they replicate that whole body response. But obviously animals don't translate directly to humans. We have very different metabolites, we have very different enzymes and very different physiologies, and so that's really where chips can come in very handy.
Yeah, for sure. Oh, okay. So which application of organs on chips do you feel has got the most potential that has not yet been realised? So I feel this links on quite nicely to the previous one.
Yeah, I think this kind of feeds into the question of the future of chips as well. So there's so many different places where chips could come in handy, but I think the thing that I'm really excited about is the concept of using them in clinical trials areas. Like you referred to, if you can get them into a hospital lab, and you can start running tissue chips from patient biopsies that you've just taken, and you do some differentiation and some IPS technologies, and you can create a linked system that will replicate the disease pathology that they're showing, and then start testing drugs in them so that you're not testing them in the patient and potentially making them quite sick. I think that in itself would just be phenomenal, and I think that's a really exciting area that we should be aiming for.
No, it would be. It's absolutely fascinating. It really, really is. And I mean, if I can add to that, I would love to see, and maybe you can make this happen. I would love to see some sort of benchtop instrument that could also do genomics type sequencing on samples. So this is a completely automated workflow, and then you can get this real time transcriptome or whatever it is that you want to look at, it's just so minimally labor intensive. So I get that in every hospital. That is, I mean, the number of lives that would be saved alone, the number of time, the amount of time that would be saved, it's just, it's such a fascinating way to
absolutely
go.
I mean, think if you had that kind of technology and you could take it out to a rural hospital in Africa. You could start understanding rare diseases in ways that you just, because people don't have access to healthcare, then what are they supposed to do? So there's so many opportunities for that kind of technology, and yes, we're very interested in how we can make that kind of thing happen.
Definitely. And you're also very interested in how we can do this in space, and this links beautifully into the next section, and this is what I've been so excited about to discuss with you, because obviously I've read the literature, I've seen your papers, and well, actually, let's just start really simply. How big is the biological research in space field? Because I imagine it's a little restricted right now.
So let me preface it with saying that anything that's done in space is under NASA's purview, right? But there is a national lab. There is a national lab up on the space station, and so that's where NIH has been working with NASA and the ISS National Lab to do some research. So everything that's going to come after this is, is sort of, thanks to NASA, because they're letting us use some of their facilities. But I mean, in the general sort of the question you just asked about how much biological research is being done in space right now, the answer is actually a lot.
Okay.
There's a lot of facilities up on the space station for lab research.
Would you mind, what can you give an overview of that? Because I'm
so
not
NASA
sure
are definitely the right people to give a better answer than me. So if I screw up any of this and anyone from NASA is listening, then I apologise. But there's some great lab facilities up there. There's some microscopes. They can do quite a lot of cell culture up there right now. They've got all kinds of plant biology that's going on. They have some ultra cool physical sciences and chemistry type experiments that are going on with things like the Cold Atom Lab. But on the life sciences side of things, they've got they can do gene sequencing now, so they can sequence bacteria that they find up on the space station.
That's cool.
So, and they've got all the basic lab facilities, freezers and all of that kind of thing, which is difficult to do in space because just the way that the air convection goes is very different. So, their whole kind of everything that they do in the lab up there is very, very different to how we do it down here. I mean, apart from anything else, imagine if your pipette floated away every time you let go of it. So,
I mean, how would you even because in space there's no gravity. Can you transfer a liquid using a pipette? Does it work as such, or
it does? Yeah, it does.
Yeah.
And so there's a lot of the astronauts who've been up there are actually scientists, and they're trained lab scientists. So we love them a lot. But they've done a lot of work in really just kind of showing how you can do research as you would normally, but there are certain things that are lacking. So buoyancy is different. Convection is different. There's definitely different things that things happen differently, but that doesn't stop lab research happening. It means there's a lot of Velcro involved so that things don't float away and you can stick them down. But Kate Rubins, who is she's actually an infectious disease scientist by training, and she was actually showing that you can pipette on a 96 well plate without it needing to be enclosed, because the surface tension of the liquids is actually enough to keep it in the well. So cool. It's just there's just really interesting things that happen, but it doesn't preclude any kind of lab research being done that we could do down here.
Okay. Well, I mean, potentially stupid question, but I feel like it's also one that needs to be asked. Are there any facilities on Earth that can successfully mimic the environment in space?
Yeah, that's harder to do. So there are certain ways that you can model microgravity here on Earth, and they're for different amounts of time that you get kind of that exposure. So there's these things called drop towers. And the clue's in the name. It's literally a tower that you drop something down. But when it hits free fall, that actually mimics microgravity just for a few seconds.
Sure.
There's also there's different kinds of machines called rotating wall vessels, or they've got clinostats is another name, and they're essentially ways that they can, for small samples or even for plants or seeds, they're ways that you can essentially rotate something kind of gyroscopically, and it reduces the gravity vector. Basically, it kind of cancels the gravity vector, pulling it down out. So it models microgravity to a certain extent. It doesn't do a fabulous job, but it definitely recreates some aspects of microgravity. So there are some ways to do it here on Earth, but it's not like we can just go into a room and press a button and then gravity disappears, because that would be amazing.
Doesn't work, no.
There's also the things called the vomit comet, which is the parabolic flights.
Okay. No.
So this is another way to model microgravity. It's basically a jetliner goes up and then it literally just does a series of humps and bumps. So it'll climb and you get a little bit of hypergravity, and then it will go over the top, over this parabola, and you get about 20 seconds of microgravity during that time. So that's actually a way that anyone who works in the microgravity space, with NASA and also private companies, they do a lot of the testing of their equipment that might go up to the space station on these parabolic flights, because it gives them a certain amount of time to be able to see how their platform works, how their system works in microgravity. It's also a really great way to get people exposed to microgravity and do kind of physiological experiments as well, and find out how they respond to microgravity. But like I said, if anyone from NASA is listening and I bungled all of that explanation, then I apologise.
I'm sure you didn't. But just very quickly, what's the difference between microgravity and sort of no gravity? Or is there no difference?
You know what? Again, I am not the right person to explain this because that definitely goes into the more physical realm.
Okay.
But my understanding is that on the space station, there is still small amounts of gravity because we're still very close to Earth. So the space station is actually only like 250 miles away. It's just going really, really, really fast. And the fact that it's going so fast, 17,500 miles an hour, and the fact that it's so close to Earth, it basically means it's in a state of perpetual free fall. And that's what the term kind of microgravity is in that respect. I think true microgravity, I hesitate to speculate in case I screw it up, but I think true microgravity, you need to be really far out and far enough away from a planetary body that you would feel that kind of gravitational pull.
With you, okay.
So low Earth orbit, which is where the space station is technically microgravity, but it's also technically just endless free fall, which also sounds really exciting.
It does. I suppose you can experience that a little bit with parachute diving, right? Like is that the same sort of gravity, or is it when you hit that terminal velocity, is it
terminal
similar?
Velocity,
like that?
Yeah. So I am actually a skydiver, so I'm very familiar with that.
Oh, okay.
It's very different when you're skydiving because you have a lot of air molecules in the way that are smacking you in the face and all over your body as you're on the way down, so you get a lot of drag. So it doesn't feel like falling, but it also doesn't feel like floating. So it's a bit different from the feeling of microgravity, I'm sure. I don't know, I'll have to go up and do some parabolic flights or try and get myself up to the space station and find out.
I was going to say, if you can, get yourself a ticket right to the space station. That'd be amazing, right? Seriously.
If I can afford like 20 million or something for one of these new Blue Origin type seats that they're selling.
Can you not justify it on work expenses? Or?
I should try. See if the US government will allow me to do that, yeah.
Okay, so can you give us an overview of what the different risks of being in space are and what effects it actually has on different organs in the body?
Yeah, so in terms of astronaut health, it's quite different to how NIH thinks about using space and microgravity, but certainly for astronauts, when they go up to the space station, there's a bunch of really interesting and not necessarily wonderful changes that happen to them when they go up to the space station. So first of all, they immediately start losing bone density because there's no gravity. They're not feeling that biomechanical pressure on their bones, and their body immediately starts actually dissolving the bones. And so there's this really quick loss of bone density. The way astronauts mitigate that is they have to work out for like 2.5 hours a day. They do a lot of resistance exercises. They have a treadmill where they're held on by bungees, so it like pulls them down to like 70% or something of their body weight. So they still kind of have that like, compound kind of aspect that helps them retain bone density. They start losing muscle mass really quickly as well because all of a sudden they're not having to fight against gravity. So they're just kind of, floating around and it's wonderful, and there's no kind of resistance for them, so they start losing muscle mass. There's also a shift in the fluids in their body. So when you're down here on Earth, a lot of the heart works really hard to pump fluids up from the bottom of your feet because it's they're down there a few feet below your heart, and so your heart's very used to pushing really hard to keep pumping the blood around your body based on gravity. When you get up to the space station or out in space, there's a readjustment of how your fluid is distributed in your body, basically because your heart is now kind of working differently and pushing the fluid around differently. So that's sometimes where you see astronauts with kind of puffy faces because they get a lot of fluid up in the tops of their heads instead that's normally drained by gravity. And also they get what they call chicken leg syndrome because, again, because the blood's not pooling in their feet, they get kind of their legs look a bit skinny. So there's this fluid shift. Cool. That can place actually quite a lot of strain on the heart. So, there's cardiovascular changes that can occur. There's really interesting changes to the eye as well, which we don't fully understand yet, but there's changes in the shape. Yeah, so I think it's to do with different kinds of cranial pressures. I'm sure it's to do with, there's lots of other more complicated reasons, but they call it space-associated neuroocular syndrome, which is basically where kind of the the function of your eye changes slightly, and it doesn't necessarily revert to normal when you come back down to Earth. So pretty much everything that happens to astronauts when they come back down here, it reverts to how it was before, for better or worse. But some things don't, and SANS, as it's called, is one of those. Radiation exposure is a big one as well. So down here on Earth, we're very nicely shielded by the Earth's magnetic field, but up in space, there's less of that, and you get, sort of more exposure to different kinds of radiation. And then there's changes in the genomic level as well. So the twin study, which was done a couple of years back, was this study of Scott and Mark Kelly, who were identical twins, both astronauts. Right. Okay. What a family, eh?
Yeah.
And they did a study where one twin went up to the space station. He was up there for a year. So Scott Kelly was up there for a year. And his brother, Mark Kelly, stayed down here on Earth, and they took samples of all kinds of, skin and blood and bodily fluids over the course of the year, whilst Scott was in space versus whilst Mark was here on Earth. And then they were able to compare them and look at, all of the kind of omics responses as well, and see a lot of changes actually in Scott's genetic makeup that reflected how his body was compensating to microgravity. So if you want to read the details, because I'm not the right person to talk about the twin study, but if you Google the twin study, then there's some fabulous papers that came out in, I think, Science last year or the year before that detail all of these amazing changes. Yeah, all of these amazing changes that happened to Scott and how they differed from what happened to Mark here on Earth over the course of a year.
That's honestly fascinating. Okay, wow. So back to organs on chips then. That's such a good overview. Thank you. But then, what chips have you sent up to space, and can you share any details of chip experiments that have been completed in space?
Yeah, absolutely. So the reason that NIH sent chips to space was not to look at astronaut health, and that's not because we don't love our astronauts.
Right, okay. But I think I just assumed that. Sorry, yeah.
But NIH's mission is to understand human health and disease, right? Mm-hmm. So astronauts are humans, so we want to understand their health and disease. But we're really focusing more on the other 7 and a bit billion of us down here on the planet who suffer from diseases, and we want to improve their quality of life and everything. So the reason that we partnered with NASA to do anything with chips in space was because of these changes I talked about that happen to astronauts. They look really similar to a bunch of different kinds of diseases and aging states that happen down here on Earth. So that loss of bone density is just like osteoporosis. That loss of muscle mass is sarcopenia, which is an age-related muscle wasting, which is a big clinical problem. Yep. These changes in cardiovascular tone, and how the heart muscle responds. There's obviously heart disease is a big problem down here on Earth. Astronauts can get kidney stones with a greater frequency. And so obviously kidney stones are a big problem down here on Earth as well. So we actually are using the environment of microgravity as a way to model diseases on a really accelerated timeline, because these changes happen really quickly when astronauts go up there. So, the extension of thinking was, well, they're going to happen to the cells really quickly. And so if we can look at, say, the formation of kidney stones over the course of days that could take decades to happen here on Earth, then we can start understanding diseases and looking at diseases in a way that we are not able to do here on Earth. So that is the reason why NIH actually is doing anything in space, is because we're hitching a ride on the space station to try and understand diseases better, and then potentially test drugs so that we can see, does this drug react up here that would suggest that it would work down here on Earth, but over these much longer time spans. So we partnered with the ISS National Lab, and again, massive thanks to NASA for helping us get up there, and SpaceX as well. But we are looking every experiment that's being done up there is has a direct translational relevance to clinical problems down here on Earth. Okay. So we have chips that are looking at heart tissue and cardiomyopathy. We have chips that are looking at the blood-brain barrier, which prevents various things crossing this kind of biological barrier to harm neurons. And so we're looking at how that's regulated in microgravity. We're looking at the immune system and aging of the immune system, or immunosenescence, looking at how T cells differentiate. So T cells are the kind of memory cells of your immune system, your adaptive immune system. And so we're looking at how the omics of those cells change in space and what that might mean for aging here on Earth of the immune system.
So are you able to do the omics in space, or is this something that you need to bring samples back down? Okay.
Yeah, so we bring samples back down. So like you said, maybe in future we can do that. But we are currently not. If NASA has those facilities, we're currently not using them up on orbit. We're bringing samples back down for that. Okay. But yeah, we're looking at muscle wastings. We talked about sarcopenia. We're looking at post-traumatic osteoarthritis. So arthritis is kind of a big problem with aging down here on Earth. But, we're looking at how that might be accelerated in space and what drugs can be used to help prevent or reverse those changes. So we've got a real broad range of chips that we're looking at. We've got chips up there right now. Some experiments finished not long ago looking at a kidney on a chip. System, which was, like I referred to, actually looking at the formation of kidney stones. So the microcrystals that start forming at the beginning of a kidney stone, and looking at whether that's accelerated in microgravity, and therefore how drugs can affect the formation of those microcrystals right from the get-go. So, and the effect it has on the cells in that system. So we've got a really broad range of chips that are up there, and they're all doing something that's very important clinically down here.
So what have you found so far? What have they told you, these experiments?
So I won't scoop any of our researchers if their results aren't published yet.
Of course, sorry. Yeah,
but I can say that we are seeing some microgravity associated changes in some of these platforms. And so what that means is it's really showing that microgravity is affecting the tissues on these platforms, which in itself we weren't sure about. We were like, we can send these chips up, but maybe they won't be bothered by microgravity because they're functioning on such a small scale, relevant to, sort of different to a full human body, that they might not care. So it's quite a relief actually that, we are seeing some microgravity associated changes.
Yeah,
so it's really suggesting that there is an opportunity to model diseases and understand biological mechanisms up there in space, which is tremendously exciting.
And then
we're also and then we're also seeing how microgravity is causing these changes, but it's going to be opening up avenues for different targets, drug targets. So it's going to be showing us potentially new pathways of how microgravity causes these changes that would open up what we might call druggable targets. So areas that we might think, hey, we didn't see this down here on Earth, but it's relevant to this disease pathology. So this is perhaps a new metabolic pathway that we can look at, or a new, protein on the membrane that might do something that we should be looking at. So we're expecting that a bunch of research papers will be coming out from our researchers in the next couple of years. Obviously research takes a long time here on Earth, but when you're talking about only being able to hitchhike to the station, once every couple of years, then it obviously takes a lot longer to get the full set of results that our teams need. But definitely preliminary data is showing that we're seeing some kind of effects of microgravity on our tissues. So
that's
you'll have to
wait
for the papers to come out.
Yeah, of course, and I will read those papers with such enthusiasm as well. I mean, it's so fascinating. It really, really is. I wondered about aging as well. Does microgravity have an effect on aging? A lot of the diseases you mentioned were sort of age associated on Earth. So is that a thing that astronauts lose a few years of their life because of being in space, or is that because of accelerated aging? Is that a thing, or have I got that?
Yeah, no. So to a certain extent, there are effects that could influence aging, but I don't think anyone would say that going up to space knocks a few years off your life. Okay. There's just not enough data to even look at that yet. And there's so many things that compensate for all the things that happen in your body. Yeah. But referring again to the twin study, one of the things that they did show is that Scott's telomeres I can't remember if they shortened when he was in space, I believe. And the telomeres are on the ends of your genes, and they can be modulated by epigenetic effects. And I'm not going to mangle the details on that, but essentially the changes that they saw in Scott that suggested that he was showing signs that were seen more in aged folks here on Earth, should we say, markers that were associated with aging, should we say. They did see in Scott, but they also saw that they pretty much reversed when he came back down to Earth. So it seemed to show that, you know, if there were mild effects on his physiology when he went to space, you know, on the genetics or on his genes, then they were pretty much generally reversed when he came back down, which I think is reassuring for anyone who's interested in going to space. It's not like they're suddenly going to turn into, you know, an old lady or something. But
it's reassuring, but it's also fascinating, because if there's some sort of epigenetic response, like you said, you know, in addition to the genes, then surely there's a potential way to block the addition to the genes, and you can delay aging, right? So
exactly.
Maybe this holds the key to, this is the fountain of youth, is it?
maybe. It's certainly a little puddle in which we can start doing some experiments. So
amazing. Yeah. Okay, again, I've got thousands of questions, but one that I really wanted to know was, what are the technical challenges associated with carrying out chip research in space?
Oh man, so many. So, you know how we already talked about how here on Earth you need a bit of a chip whisperer sometimes to get your systems working, because you've got microfluidics, you've got pumps, you've got tubes, you've got valves, you've got incubators, you've got refrigerators, you've got cell culture, you've got sterility techniques, you've got all of this stuff. And then in a lab, that can be hard enough. But then if you think you've got to shrink it down to something the size of a shoebox that you can put on a rocket and blast up to the space station, you have to make these automated, self-contained platforms that are so robust that, you know, you've got to keep your cells alive. Even just keeping the pumps going and keeping the cells fed can be so challenging. So the technical adaptations that have had to happen for these platforms is absolutely unparalleled. It's so difficult. And so all of our teams that we're actually funding, they have partnered with what we call implementation partners or payload developers. These are experts in the space field who know how to get a mechanical system plugged in to a Dragon capsule and, you know, keep it going, get it plugged into the space station and you know, make sure all the electrics plug in and they haven't put, you know, a UK plug on a US socket, that kind of thing, you know.
Imagine.
Oh God, yeah. But that in itself is incredibly complicated and requires deep expertise in the space field. So all of our teams have partnered with companies that are very skilled at doing this. But then they've also had to shrink down all their systems. So they've had to miniaturize them, they've had to automate them, they've had to make them very robust. And those are not trivial challenges.
Oh, no, definitely not.
They've also had to make them straightforward enough so that, and this is not our words, this is directly from the words of the astronaut Terry Virts, that they are astronaut proof or pilot proof, because a lot of astronauts were pilots as well. So they have to be so simple that someone as stupid as an astronaut, and again, his words, not mine, but someone as dumb as an astronaut can use these systems. So they have to be very simple and straightforward to use as well. The astronauts do get some help from, you know, an angel sitting on a body cam and someone talking in their ear to help them talk through some of the procedures, but definitely the technical adaptation for these platforms has just been so difficult, and just it's been phenomenal to see what our teams have been able to achieve.
Wow. Yeah, I mean, I've had to build my own systems for doing organ on chip, and I mean, miniaturizing it and making it, it's just such a minefield. There's so many things that can go wrong, like you said. It's absolutely it's crazy. So how throughput are these systems that you're taking up? How many experiments can you typically run in one payload?
So it depends on the chip, it depends on the platform, but we have replicates up there that we had 24 kidney chips, I think, that are up there right now, but they're triplicate chips. So they're actually able to get 72 replicates, biological replicates, which is pretty high, actually, as far as tissue chip technology goes. We've had, I think, some of the experiments we've got coming up will have 96 replicates going up. So we'll be able to get really high biological replication, the number of samples that we can run at any one time. That's relatively unusual, I think. For most chip experiments right now, they're pretty low throughput. If you can get maybe 6 or 12 running at the same time, then that's seen as kind of high throughput. And it's partly because, like you said, they can be very complicated to set up. You've got to get them all sat in the incubator with the pumps going at the right time and, all this kind of thing. So, to get as many as, 72 biological replicates within, a tiny system the size of a shoebox is just really remarkable.
It is. It's fascinating. And it shows as well that all of this research is not just biological. You really need all of the interfacing of all of the different disciplines. You need the
Absolutely.
robotics engineers, you need the mechanical engineers, all of this sort of people. And it's very difficult to be able to even, to get all of those people working together because everyone has such different ideas about things. And so
I work at the National Centre for Advancing Translational Sciences at NIH, right, or NCAS. And everything we do is exactly what you just said, which is pulling together all these people from all these different disciplines, because we're looking at things, we're trying to ask questions and answer questions that need all those different backgrounds to be able to address these problems. We call it, it's translational science, but it's really team science. It's really bringing together people with really diverse backgrounds, trying to get them all on the same page, and then hopefully trying to inspire them and get them excited about what they're trying to do, which certainly with Chips in Space is easy because everyone's very excited about that.
Yeah, for sure. So, okay, are there any chips that are easier to work with than in space than others?
I would say none of them are easy because there's all the reasons we just talked about.
Yeah.
But I would say that the ones that don't involve flow, so that don't necessarily need pumps running 24/7 to keep the cells alive and might just need a media exchange every few days.
Sure.
I would argue that those are a little bit more straightforward just because the engineering needed to keep them going is marginally less complicated.
Yeah, makes sense.
But I would say that, really they've all been, very difficult to get off the ground, literally.
It's so fiddly to do it down here on Earth, that manipulating all of the fluids and the droplets and all of that. To do it in space again when the droplets are misbehaving and floating off, that would be an absolute nightmare.
Well, NASA's very careful about their biological containment. They have 3 levels of containment for everything. So we're certainly we have not heard of any of our samples that have escaped and gone floating around, but I couldn't possibly comment on some of the stories I've heard about various things floating around on the space station in the past. So.
Yeah, I'm sure you can make a whole other podcast episode on this.
Yeah, exactly.
Okay, so to round it off, because again, I've got 1,000 questions, but we have to finish at some point. So what's the future of the Chips in Space program?
So the future of the Chips in Space program from our perspective is that it will be winding down over the next couple of years. We funded all of our teams to do 2 flight experiments, and they have all done their first flight, and most of them are now coming up for their second flight. So once their second flight is done, they'll be analysing their data and putting out their publications. And from our perspective at NIH, we're not ruling it out, but we don't have any big plans right now for additional programs with Chips in Space. That said, we've worked with so many people who have been so supportive and really interested in the technology that I fully expect that tissue chips will remain in the space realm in some form or another over the coming years. Just because of all the things we've talked about, there's so many opportunities to understand disease modelling, but also for, with precision medicine, like we talked about, to understand maybe astronaut health. Maybe start understanding the different risks that astronauts are associated with or that will be exposed to on long-term missions to the moon or further afield. I would really love to see chips used in that context. Maybe there'll be chips on the moon in future, who knows? So we'll see how that pans out. So it's an exciting future. And down here on Earth, there's still so much work to be done. for tissue chips. We've got 5 or 6 different programs that just NCATS runs. There are other programs across NIH where different institutes and centres at the NIH are funding chip research. There are different government organisations that are running their own programs. There's so much work going on in the private sector, the pharmaceutical industry. That there's just a real explosion of chip work down here on Earth, and I think we're going to see some really exciting advancements over the next few years.
Yeah, I couldn't agree more, and especially when you said about the long-term space journey. I mean, for colonizing other planets, like has been talked about by obviously Elon, and that tissue chips can play such a huge role in that, and sort of like trying to understand the effects of long-term space flight on, like, or even like trying to figure out how to do the you see those cryogenic pods in all sort of space movies that people are in, right? All that kind of research, that's tissue chips have got such a huge role. So I mean, it's such a fascinating space.
And I would just jump in and say that the one thing that we always try and point out is that the promise of tissue chips is super exciting, but it's going to take a while to get there. It's early technology. Like we talked about some of these challenges. These are difficult challenges, technical and biological and scientific. So it's really going to take the whole community, like we talked about those transdisciplinary teams to address some of these challenges. And also tissue chips, even if you create let's say a perfect system that is self-contained and automated and, can be used anywhere, it's still only a model of some cells in your body. It's even if you link a bunch of them together, it's still only going to be able to model certain aspects of your body and certain responses in certain contexts. So I think we always say there's so much promise for them, but we also have to remember that they're just one piece of the puzzle. So there's going to be lots of other tools that people need to use to still address these questions and find answers to these things. So we're excited about what tissue chips can do, but we also don't say that they're going to be the panacea of everything biotechnology and that we're suddenly going to be able to do everything on chips.
Yeah, definitely. But like you say, it's one of those things that they've got so much promise, and the alternatives to animal experimentation in terms of animals and humans have just fundamentally different biologies. That is going to be such a huge thing in the drug development processes.
Absolutely.
But then a lot of the sort of the comments in literature right now are things like, are tissue chips over-promising and under-delivering? And I mean, what see, I knew I should have stopped asking, but what do you think on that? Like, what do you think?
I think it's a really fair concern. I think it goes back to what we were talking earlier on about the importance of defining the context of use.
Sure.
So if you're creating or developing a platform that is phenomenal for modelling bone cancer and looking at different drug therapy combinations, and you can link it to a liver system and you can link it to a kidney system and see the off-target drug effects from this particular combination chemotherapeutic, then you've got potentially a really nice system that you can define the boundaries, you can define the questions, you can define very specifically the questions you're asking and how you get those answers. I think because it's new and because everyone can see the promise, tissue chips are vulnerable to the hype, effect. You'll see something come super hyped and then it'll all kind of come crashing down and it just won't ever really get taken up. So that's why there's so much focus on almost kind of scaling back the excitement a little bit and being like, right, what are the tangible things that we need to address, like validation, like cell sourcing, like, robustness and reliability. And how can we do those things to build confidence? We're doing a lot of things at NCATS to try and pull together information from all different kinds of tissue chips. We have a database, an MPS database, where we're really encouraging everyone to put their data into it. And this database has amazing computational and analytical tools that can help you see exactly how reliable your platform is with different drugs, at different places. So again, like we were talking about chip whisperers, chip whisperer at lab A and chip whisperer at lab B, do they get different results doing the same thing? So we're funding this database that is pulling in all of this data that is really helping answer some of those questions to help build confidence in the system, so that it's not going to be subject to that, that when that pipe bursts, that the technology won't just kind of disappear. So we really want to help sustain the interest whilst tempering the enthusiasm with the realities of what is possible right now and what still needs to be addressed.
That's so cool. Thank you. I mean, that was such a wonderful overview of everything. I really, absolutely fascinated. In the next section, I want to hear all about your career and how you've got to where you are. But for now, thank you for that. Cheers.
No, you're welcome. That was a great discussion.
So for this next section, I want to hear all about your career path, and it's called When You Were Young. So where did you grow up? Where are you from? And what did you study as an undergraduate?
So I am an Essex girl and proud. I grew up in Chelmsford, Essex, and I certainly never envisioned myself where I am today. But I was very lucky. I went to a very good school in Chelmsford, and then I was lucky enough to get into Oxford. As an undergraduate, I went to Magdalen College, Oxford, and I studied physiology with psychology, which was the closest I kind of got to neuroscience at that stage. And the reason I kind of went for that is I was always a big biology nerd. I couldn't decide between biology and geology and geographical sciences, and sometimes I think I made the wrong choice because I could be hanging out by volcanoes if I'd gone, down the geology route. But I went to a lecture in London at the Royal Society, and it was by a female neuroscientist, and she was absolutely phenomenal. And she was talking about the brain, and it was like a light bulb went off in my head, and I was like, that's what I want to do. I want to do neuroscience.
Oh, okay, cool.
So that was where I kind of decided to focus my energies. So I went off to Oxford, very grateful, had a fabulous time. And then I went to University College London and did my master's, and then stayed there to do my PhD as well. And I actually worked on pain research during my master's and my PhD. So pain is very translational in nature. You think about everything from the cell to the human, and everything in between. So you have to kind of understand all of these different mechanisms. So I was actually looking at the long-term effects of pain during childhood, and the neonatal period, and how it affects reward processing in the older animal in this case, but also directly translating to humans. So I was really interested in the kind of the pain reward spectrum, which was very prescient because of, certainly in the US now, the opioid crisis over here. So,
yeah, of course.
So that was fascinating. And then I was very...
So what did you find out? Sorry, what was the outcome?
So I found out some really subtle effects that a repeated neonatal pain, and in this case it was a foot wound in rats. It was like equivalent to a heel lance in neonatal babies, and they have a little knife that goes and hits the heel to take blood, to check that their blood oxygenation levels are high enough, but they have it done a lot. And it's a little bit similar to a skin incision injury in animals, which is used quite broadly in the pain field. So I found that a repeated foot wound in neonatal rats actually very subtly affected their responses to food reward in adulthood. And in this case, I was using Cheerios, breakfast cereal, because rats love Cheerios.
But I was putting them in... It's like chocolate.
Yeah, and I was putting them... Exactly. Oh, they love chocolate. But I was putting them in like an aversive environment. It was like a bright white environment, which they had to like cross to get to the Cheerios and take the Cheerios. And I found that they didn't... that they loved the Cheerios just as much, but when they had been repeatedly injured as little baby rats, they were a little bit less willing to go and get the Cheerios. So it was a very subtle effect. It would have been fascinating to carry on that work and really kind of get into the neurobiological underpinnings of it, which I just kind of managed to scratch the surface during my PhD. But after that, I went to Montreal. I went to McGill University in Montreal and was looking at chronic pain. So looking at the effects of chronic pain and how it changes brain structure and function. and also how emotional and environmental factors can affect that chronic pain and the neurobiology underpinning it. So I was working with a fantastic mentor and PI called Catherine Bushnell, who is a brain imaging expert, and I was doing all these crazy rat experiments and rat brain imaging, and so that was lots of fun. And then she invited me to join her down at NIH in the US a couple of years later, and that was definitely an offer that I couldn't refuse to go to NIH. So I went to NIH in 2012, and then I transitioned over to the tissue chip program. So from neuroscience to bioengineering, if you like, about 5 or 6 years ago.
Okay. That's awesome. So, I mean, just going back to all of these sort of thought processes that you went through, you said it was a no-brainer to move to the NIH. I mean, fair enough. But in terms of when you were looking to do your PhD or your first postdoctoral position, did you consider alternate career paths, or was it just that you had in mind exactly what it was you wanted to do, and you were just gonna do it? Sort of why did you decide to do any one particular thing?
Yeah, so I think, like, I remember that lecture I told you about was very pivotal, when it was just like, wow, neuroscience is just amazing. And so I went off and did a master's before I did a PhD, because I wasn't sure how I was going to get on with research. I didn't know what to expect, because I hadn't really done it.
Yeah.
So the master's was really a way just to kind of have a go in the lab and see what happened. And it was really fun. And I was like, this is so cool. There are so many techniques and labs and opportunities to just do fascinating research that I was just really excited about doing a PhD. And I was very lucky to get a Wellcome Trust scholarship to do it, to be down at UCL. So, you know, I think without that, it would have been a different story. But I was very, very lucky and grateful to be awarded one of those. So nice. I was able to spend 4 years pottering around the lab and you know, doing everything from electrophysiology to animal behaviour to clinical studies and human studies. So really interesting, exciting time.
Yeah. So then when you were finishing up with your PhD, what was sort of like your thought processes? You definitely wanted to move abroad, or because obviously you live abroad now, expat living abroad. But you know, did you ever consider staying on in your lab? And why did you want to move to this particular place? Was it because of the researcher that was there? And then how did you contact them? You know, all of these sort of
yeah. So no, that's great questions. The thought process was that in science, and when you do a postdoc, even if you choose not to stay in science, a postdoc is a phenomenal way to go somewhere completely different and live somewhere else for a few years. So I was really excited at the prospect of doing that, and I looked at a bunch of different labs that were doing really cool research that I was really interested in. There were some in the west coast of America, there were some up in Canada, on the west coast of Canada, and then I was actually running a seminar series, and I'd invited Catherine, who ended up being my PI, to come and talk to us in London because she was doing such cool research. I was really interested, and I was like, well, if I'm running the series, I get to invite her. So I invited Catherine over, and she was talking about the research, and I was like, that is amazing. And I was saying to her afterwards, I was like, Catherine, that is so cool. Can I come and work with you? And she's like, yeah, sure, but you need to find your own money. Ooh. And that was something that a lot of the PIs were saying, was like, we'd love to have you, but we don't have any funding, so you've got to find it. Yeah. So that's when I started applying for a lot of different fellowships, a lot of funding opportunities, and I was very, very lucky again. I applied for one through the International Association for the Study of Pain, or IASP. It was the John J. Bonica Trainee Fellowship. Oh, and basically, okay. Catherine and I found out about it a couple of days before the deadline, hustled together an application for the project I was going to do, and we put it in, and I was very, very privileged to be awarded that in 2010. 2009, I think it was, but then that enabled me to go to Canada in 2010 because that actually covered a salary and some lab fees. So Catherine was like, brilliant, we got some money, come on over, come join the lab. So I was able to go over to her for a couple of years on that fellowship, and then luckily, you know, the next thing came along while I was there. But there was definitely periods when I was like, oh God, what am I going to do once my fellowship runs out? You go back to the UK. I was thinking at one point I was going to give it all up and go to Australia and join the circus. You know, definitely sort of worrying about what I was going to do next. Oh yeah. No, I get it. It's really tough. It's really tough. Yeah. And one of the things that when I got to NIH, I got very involved with was the Fellows Committee. So advocating for and representing fellows, postdoc fellows at NIH and across the country, because postdocs are really the workhorse of the lab. They do all the work. They're the most experienced people in there, other than the PIs and some of the lab techs who've been around for so much longer. But they're really the things that the people that make research happen. And I feel like they're just, certainly in the US, pretty undervalued. And that annoyed me, frankly. So I wanted to sort of stick up for them and speak up for them where I could. And I was very lucky that NIH is actually very supportive of its postdocs and its trainees. So it was a really good environment to be in.
Cool. Okay, so to round out this section, what's your golden advice that you'd give to sort of postgraduates or post PhD? So either in postdoc, or if you're working in industrial science in some sort of capacity. What sort of advice would you give to people if they're sort of at a little bit of a juncture and they don't really know what to do?
So from the pre-PhD level, I would say if you're at the PhD level and you're working on it, don't give up. Because once you've got it, they can never take it away. And you'll have that little warm, fuzzy doctorate just sat in your heart forever. And trust me, some days that's what gets you through, is like, once you got it, they can never take it away. So it really is an achievement to complete a PhD. And so I think it's very easy to forget that when you're in the throes of it or when you're thinking about it. But I would not have changed my PhD experience for the world, as terrible as it was at times, but ultimately massively fulfilling. Once you get past that PhD stage, I think it's really important to bear in mind, certainly in the life sciences and in the biological sciences, that the job market is tough. There are not many positions for academics.
No, and it's very competitive.
I think, one thing I really struggled with, because I always wanted to have my own lab and be a PI, and I did a postdoc for 6 years in the end before I was like, I don't think this is going to happen. But I think it took me a long time to stop beating myself up over that and feel like a failed neuroscientist. And I think that's something that people with or PhDs, they tend to undervalue themselves. They don't realize the transferable skills. I know everyone talks about transferable skills, but really the sort of the experience that comes with doing a PhD and the critical thinking and the scientific design and all of that gives you skills and experiences that really make you stand out in many, many areas. And so I think it's very important for people not to feel like a failure if they do stay or choose to leave the academic pipeline.
Totally. Yeah.
But I think those who stay in the academic pipeline should enjoy it. Yeah. Where they can. I know it's tough because I work, having gone from trying to get the funding, I now work on the other side where we, work with the people who we're giving funding to. So I really get it. But, I think the key is to not lose sight of the science that you're doing and why you're doing it, even because it's easy to get bogged down with the administrative crap and all that kind of thing, but try and sort of take a step back and see the perspective of it sometimes and remember why you were excited about it in the first place.
That's wonderful. You put it so well. I mean, I think we're sort of fed the idea that being a professor is the be all and end all if you've gone through being a PhD, and it's quite difficult to realize what the alternative career paths are. And that was also one of my motivations for starting this podcast, hopefully like giving people different ideas about what can be done. And you've been such an amazing guest to be able to shine light on that. You're sending organ on a chip into space, like you're enabling all this amazing research that's going on. You're involved in it at such a high level. You don't have to be this person who's like doing the administrative stuff. It's
exactly. And I mean, a lot of my job is administrative. It's emails and it's meetings, especially in the US government. There's a lot of bureaucracy. But it's being able to go to conferences and talk to scientists and read papers and, stay connected to the science, which is so exciting. that sometimes makes the rest of it worthwhile. And in every job, there's always the bits you don't enjoy. You know, when you're in the lab, you still got to clean up afterwards, and that doesn't feel like bench research. It doesn't feel like you're doing anything useful, but you still got to wash the beakers. You know, so it's the same thing. You know, you've got to take the rough with the smooth in any job. But I think it's really important to work out what works for you as well. So if you are excited by thinking about the broader environment of science, or if you're excited about communicating science, or if you're excited about creating new biomaterials, and you want to be hardcore, like in your thing, doing really amazing science, then I think it's just take the time to work out what you enjoy and what you think you're good at. And bear in mind that not every day is going to be unicorns and space stations and like perfect Western blots.
It never. I'm still yet to get one, to be honest. There's always a smudge.
So yeah, that's my advice.
So I'd like to get some final thoughts from you now. We've sort of talked about this a little bit already, but in 50 years' time, how do you see the future of organs on a chip?
Ooh, in 50 years' time, I would love to be able to see multi-organ linked systems that create a body on a chip, but created entirely from stem cell derived sources from you as an individual. So take some blood, take some skin, populate a whole body on a chip system. I would love for those to be able to be broadly available in hospitals, labs all over the place, on the moon, in space, on Mars. By that point, who knows? But really, essentially, reliable ways to recreate you on a chip so that you can understand how your body works in this kind of avatar system, and look at how drugs might affect it, look at how diseases might affect it. But also, get treatments that work for you specifically through those kinds of systems.
Definitely. So you have a great overview of organs on chips, but you've also worked in many different areas. So in science in general, which emerging trend, and try not to be biased, I suppose, towards organs on chip here, we are both in the same sphere, but which emerging trend in science in general do you see as having the greatest potential?
So I'm really excited right now by synthetic biology. I think that's got tremendous promise for all kinds of different applications. Some of the stuff that we're doing at ENCATS is really exciting. We're doing some sort of crazy science fiction style ideas where we're talking about understanding or creating diagnostic devices that can sense things in the air and kind of smell things in the air and diagnose you that way. So I guess development of really kind of crazy sci-fi tech I think is really exciting. And I mean, I think I'm just really excited to see how stem cell technology continues to grow. That's not necessarily like the kind of the coolest, craziest thing in the world, but just the promise of stem cell technology is so vast that I think it's going to be really exciting to watch.
I mean, we're missing a library of protocols for all the different cells in the body, like to be made via stem cells. That that in itself freely available.
Yeah, and partnering it with joining that with the new techniques in genetics, omics, and CRISPR and all of that are coming out. I think is just going to be so exciting to see how creative people are with those technologies.
Yeah, totally. So this is the very final question, and this is the series finale, and it's been brilliant for this entire podcast season to hear all of the different answers to this question. So to round out the season, Lucie, if you could do it all over again, what would you change and what would you keep the same?
You know what? I don't think I'd change a thing. I think everything I've done from the 14 to 16-hour days in the lab at the end of my PhD, when it was just like, just get through it, I would not change that for the world, because everything I've done has led to the next thing, whether deliberate or accidental, and I'm having a blast. And I'm really excited to see what's going to be coming up in the next few years.
Yeah.
And so, I don't think I'd change a thing, because it's been an absolute blast, and I'm excited about what's next.
Amazing. Well, thank you so much for your time, for appearing on the podcast. I really, really, really enjoyed that discussion, and I wish I could ask 1,000 more questions. So we might have to get you back on at some point in future. But yeah, thanks again, Lucie. I really appreciate you coming on.
Oh, no, that was wonderful. Such a great discussion. Thank you so much, Alex.
What a way to end this series! Organs on Chips in space. A huge thank you to Lucie for such an amazingly inspiring episode, and a massive thank you to all of the guests who've appeared throughout Season 1. Importantly, I really want to thank you for listening and for supporting this podcast. It'd be nothing without you, and I hope that you've been able to get the advice that you would not have been able to get elsewhere. Making a Scientist will be back for a second season later this year with more high-profile guests and early career researchers. Be on the lookout on our social media for clues. If you've got any suggestions for future guests or questions you'd like to hear answered on the podcast, please get in touch. It would be amazing to hear from you. If you can keep spreading the word about the podcast and give it a helping hand by following us on Twitter, rating us on Apple, Google or Spotify podcasts, and subscribing on our YouTube channel, I'd be so very grateful. That's it from me. I'll see you in Season 2.